Hello everyone, welcome on behalf of Science Academy. Today we are going to discuss on the topic photosynthesis. This is a very important topic in all upcoming competitive examinations and board examinations. I have decided to keep this topic distributed in different lectures so that I could deliver each and every minute detail on each and every point, and you can pick up or note down whatever you want. So let's start. Okay, let us look upon the key points which we are going to cover in this lecture. We will study about photosynthesis as a broad general term, the common definition, okay, for a nice introduction. Then we are going to study about the organisms involved in photosynthesis. Then we are going to study about the photosynthetic pigments involved in this process. And lastly, we are going to study about the location of photosynthetic pigments. So, what do you mean by the term photosynthesis? In broad or general term, we can define this process as a process in which there is synthesis of organic compounds by the aid of light energy. Okay. Now, who does it? The green plants, or specifically any photosynthetic organism. Photosynthetic organisms convert this light energy into chemical form of energy, okay, which are in the form of NADPH or ATP, and this is further used to reduce carbon dioxide to form organic compounds. This process or this step is also known as carbon fixation step. We will study about it in details. Now I have inserted here a picture of the overview of oxygenic photosynthesis. This is actually a very important process, or this is in just the nutshell of whole photosynthetic process. So I have just here mentioned the overview. You can note it down and draw the diagram for your future note purpose. Okay. So from the diagram it is clearly seen that it is a two-step process. The first step is a light dependent process, or generally it is called as the light reaction. This process is a light dependent process that requires the direct energy of light to make ATP and NADPH that are used in the second process. The second process is what? It is a carbon fixation cycle step or the Kelvin cycle step, which occurs and in which the ADP and NADPH are further involved in the reduction of CO2 to N-triose phosphate. Okay, clear? If you have any doubts, please comment below. Now we will study about the photosynthetic organisms involved in photosynthesis. There are different types of organisms involved in photosynthesis, ranging from photosynthetic bacteria to long, large green plants. These photosynthetic organisms may be oxygenic or anoxygenic. We have divided this on the basis of generation of oxygen during photosynthesis. Anoxygenic organisms are those which do not produce oxygen, okay? And oxygenic photosynthetic organisms release molecular oxygen. I repeat, oxygenic photosynthetic organisms release molecular oxygen via the light dependent oxidation of water to molecular oxygen, okay? Whereas anoxygenic photosynthetic organisms do not produce oxygen. This is the basic difference between both of them. So here in that slide you can see the examples. In anoxygenic section, we know the green and the purple photosynthetic bacteria are involved, while in the oxygenic photosynthetic organisms, both in eukaryotes or prokaryotes are involved, as we all know. In eukaryotes, the green plants and any photosynthetic protist, okay, most of all of them. And in prokaryotes, the famous cyanobacteria is involved. Okay, now let us discuss about the pigments involved in photosynthesis. Different kinds of pigments are employed for capturing solar energy during this process. The major ones are chlorophyll, carotenoids and phycobilins, as I have mentioned here. There are various types of chlorophylls majorly employed, out of which I have given few examples like chlorophyll A, B, C and D. In carotenoids, we have xanthophylls and carotenes. And in phycobilins, we have physerythrobilin, phycocyanobilin, and allophycocyanobilin. Okay, please note these down in your copy. We will study about these in details in the next slides. Okay, first of all, let us study about the chlorophyll molecule. Now, chlorophyll is present in all oxygenic photosynthetic organisms. We all know this. There are several types of chlorophyll molecules like chlorophyll A, B, C, D, etc. These are lipid soluble. Okay, keep this in mind. These are lipid soluble and absorb light most strongly in the blue and red portions of electromagnetic spectrum, and it is very poorly absorbed in the green portions. Okay, it is chlorine or porphyrin derivative. We will study about this with the structure. Anoxygenic photosynthetic organisms also use chlorophyll, but these are generally known as bacteriochlorophyll molecules. They are related to chlorophyll molecules in different way. Different groups of anoxygenic photosynthetic organisms are used here. These are mentioned as BCl A, B, C, D, E. Okay. They absorb light at longer wavelength as compared to normal chlorophyll molecules. Keep this in your mind. So here I have inserted the diagram of chlorophyll. Chlorophyll, generally the chlorophyll structure of A and B, the generalized structure, and then according to the A or B molecule you can substitute the R chains. If you find this picture hazy or something, you can also download it from internet, or any other book and study. Now let us study about few points about this structure. Now what is chlorine? Chlorine is a large heterocyclic aromatic ring comprising of 1 pyrroline and 3 pyrroles linked through 4 methane groups. Okay, linked through 4 methane groups. Now, for your kind information, there is a misprint in this slide. It's not chlorine, not E, it's just CLORIN. Okay. Pyrroles, what are pyrroles? Pyrroles are 5 atom ring with 4 carbon atoms and 1 nitrogen atom. You can see in the diagram. Please take a screenshot and zoom out. Later you will see that. Most of the chlorophylls like A, B and D are chlorine derivatives. But chlorophyll C is a porphyrin derivative. What is it? It is a porphyrin derivative. Now what is porphyrin? Porphyrin consists of 4 pyrrole rings. Okay, now this is the basic difference between chlorine and porphyrin. Reduction of 1 pyrrole double bond in the porphyrin ring leads to the formation of a chlorine. So did you get what is the difference between porphyrin and chlorine? It is clear to you? Now magnesium ion is present in between the rings. We all know that magnesium ion is covalently bound to the 2 nitrogen atoms and coordinately bound to the other 2 nitrogen atoms in this tetrapyrrole structure. You can see that, okay, it is given very clearly. A cyclopentanone ring 5 is attached to pyrrole ring 3. See, there is mentioned ring number 1, 2, 3, 4. It is clearly mentioned there, and you can also see ring number 5. At ring 4, you can notice there a propionic acid group forms an ester bond with an alcohol phytol. Phytol consists of a long branched hydrocarbon chain with 1 carbon-carbon double bond. Okay, it is derived from 4 isoprene units. How many isoprene units? 4. Chlorophyll C is unusual because it does not have an isoprenoid tail. If you want to know anything about chlorophyll C in details, please let me know about it in comment section. I will surely include it in upcoming slides. Okay, so let us now study about the carotenoids. What are carotenoids? These are lipid soluble long chain conjugated hydrocarbons containing a string of isoprene residues and are distinguishable from each other via their end residues or the end groups. They are generally C40 terpenoid compounds. What are they? They are generally C40 terpenoid compounds formed by the condensation of 8 isoprene units. Please note it down. Formed by the condensation of 8 isoprene units. Okay. Now they can be subdivided into two major classes. The first one is xanthophylls, which contain oxygen, and the second one is carotene, which are purely hydrocarbon and they don't contain any oxygen. The characteristic absorption spectrum of them is 400 to 500 nanometer, and in this is responsible for the the main yellow coloring of leaves in autumn or the orange color of carrots. These pigments are mainly responsible for their color. Now these pigments act as an accessory light harvesting pigment. You will study about this in later detail. And it also acts as a photooxidative damage shielder by acting as a quencher and scavenger. If you want to know in details, I will make another video on this particularly, how it acts as a photo quencher and all of it. Okay. Now here I have inserted the picture in which there are two types of example of carotenoids. One is from the carotene family, which are devoid of oxygen. Beta carotene is the example, and the other is xanthophyll, which contains oxygen. Now the example here given is lutein. Okay. Now let us study about the phycobilins. Phycobilins are non-cyclic linear tetrapyrrole structures. They are very similar to the structure of bilirubin. I hope you all know about bilirubin. Chlorophylls are composed of 4 pyrroles, but the pyrroles are arranged in a ring and contain a magnesium ion in the center. The 3 photosynthetic phycobilins which are mainly involved in all these processes are, as I have discussed and I have shown you in the previous slide. What are they? Physerythrobilin, phycocyanobilin and allophycocyanabilin. They are the unique among photosynthetic pigments. Why they are unique? You know why they are unique? Because they are covalently bound via the thioether bond. Okay, I have mentioned in the slides or not this portion? Okay, you can note it down. They are covalently bound by the thioether bond to certain water soluble proteins which are known as phycobiliproteins. See, I have mentioned about phycobiliproteins. They are mainly of two types, physerothrins and phycocyanins. These phycobiliproteins, so the phycobilins are attached to phycobiliproteins via the covalent bond which is thioether bond. Okay. Now these proteins are water soluble, they are not lipid soluble, so when in membranes they form aggregates or clusters and they are present in that clustered form and they are known as phycobilisomes. I have mentioned there in point number 4 about the phycobilisomes. So phycobilins exist as phycobilisomes. They are abundantly found in red algae and cyanobacteria and they act as an accessory light harvesting pigment. They also act as an accessory light harvesting pigment. I have also inserted the picture of phycobilins, the general diagram, the general structure. This may appear a little bit complex to you, but if you practice these structures daily, I assure you, you will remember them by heart always. Okay, just revise daily, just draw one structure daily and just revise it. I mean, draw all the structures each daily and just revise it, or just draw all of them and make a note copy and make it a habit to look on it daily. Okay, daily in the morning or in the evening, make a time, give 30 minutes to all the structures, just keep a note on it and then proceed further with your studies. Now we will study about the location of photosynthetic pigments. Where are they located? In oxygenic photosynthetic organisms, photosynthetic pigments are found in thylakoid membranes. Yes, I have mentioned here in the slides. You can note it down. In eukaryotic photosynthetic organisms, thylakoids present within the chloroplast. In cyanobacteria, there is an internal system of thylakoid membranes in cytosols in which the light reaction of photosynthesis and respiration occur. The thylakoid membranes are found as pairs of bilayers that enclose the thylakoid luminal space. And what happens in anoxygenic processes? In anoxygenic photosynthetic organisms such as those we have mentioned earlier, green sulfur bacteria, purple sulfur bacteria, photosynthetic pigments are found in plasma membrane and vesicles which are known as chlorosomes. attached to the plasma membrane. In the purple sulfur bacteria, the bacteria chlorophylls are located in invaginations of the plasma membrane, which are what? Which are, we all know, which are known as chromatophores. Okay. So I hope this slide is totally clear to you. This is all for this lecture. We will discuss about chloroplast and all other related aspects in much detail in the next lecture. I would request you all to note down whatever is mentioned in the slides and follow your respective books. Once after going through this, I can assure you will remember all of it by heart. Good luck and good wishes for your future. We are totally new on this platform and working very hard. If this video brings any help to you, do not forget to subscribe to our channel. Do like and comment to encourage us so that we can always help you with our precious videos and lectures and encourage you to do something positive and something better in life. Thank you.